Mission Bio was a spin-off out of UC San Francisco, out of pioneering microfluidics lab, Adam Abati. And so he invented the technology, what we call the two-step microfluidics, where you have actually these cell suspensions can be pushed into this microfluidic cassette device, and then we can capture them and with two separate reactions. So the first step, in this case, we use a protease reaction where we can digest away the very tight chromatin structure that wraps around DNA. And in the second step, where we introduce cell barcoding as well as PCR reactions, then we can actually amplify uniformly the DNA sections of interest and actually do sequencing. In addition, because of the flexibility of the system, we can also introduce antibody conjugated with oligos, where we can then monitor protein expression. So that includes both extracellular protein, then we can define the cell types, we can understand the precise which cell types, as well as intracellular, where we can now understand the mechanisms of actually what is going on inside the cell. We can do all of that on the same cell. And so that's really the power of the system, where all of the other single cell companies actually have been focusing on just RNA. As you think about the paradigm of DNA being really the fundamentals of genetics, to RNA, which is the intermediate, and the protein, which constitutes the functional component of the cellular function, is really on the biology side is super fundamental for us to be able to understand the DNA and the protein changes at the single cell level.